Hello and happy Friday, everyone. Thanks for attending this talk. My name is Bryan Kennedy, and on behalf of Mesovision Lab, I am presenting our talk, Amblyopia-Related Changes in the Fine-Scale Functional Organization of Human Extrastriate Visual Cortex. Before starting, we need to inform you that among the authors, Dr. Bex has financial interest in Adaptive Sensory Technology, LLC, and a patent with Mass Eye and Ear in Northeastern University. Dr. Hunter has financial interest in Luminopia and Rebion, and a patent with Boston Children's Hospital. The rest of the authors have no commercial relationship to disclaim. As you may know, amblyopia is the #1 cause of vision loss in children and young adults. This developmental disorder is caused by a disruption of binocular vision in the early stages of life. Amblyopic individuals typically show decreased stereoacuity. Animal models of amblyopia suggest that this impairment is associated with a decrease in the number of stereoselective neurons. However, direct evidence for this phenomenon in humans is rare, due to the small size of stereoselective neuronal clusters in the human visual cortex. Moreover, it is not clear what happens to the other neuronal clusters located nearby that are involved in color and motion perception. These clusters are expected to show only a minor to no impairment due to amblyopia. So, do they expand as expected from neuronal plasticity? Or do they remain unchanged, in which case we expect to see more non-responsive regions in amblyopes compared to controls? This slide shows the organization of stripes in the dorsal parts of human V2 based on using cytochrome oxidase in a post-mortem histological study. To help you see these stripes better, we have added these dashed lines on top of them. Among these stripes, a subset of them are homologs of monkey thick stripes that are involved in motion and stereopsis encoding, while the others are homologs of monkey thin stripes and are involved in color perception. As you can see, in humans, it is hard to distinguish these stripes from each other based on their shape. The small size of these stripes also makes it hard to visualize and to distinguish them based on their stimulus selectivity in vivo using conventional functional fMRI techniques, which have a resolution close to 1 centimeter. Our study was based on 3 experiments, all of them based on using high-resolution fMRI collected in a 7T scanner, which gives us a nominal resolution of 1 millimeter. Using this technique, in the past, we've been able to localize motion, color, and stereoselective columns across early visual areas. Here, we use this technique in amblyopes and controls to localize these stripes and to compare their size and response to different stimulus conditions. Up to this day, 27 individuals have participated in our study. Among them, 7 were strabismic and 5 were anisometropic, but they all showed impaired stereoacuity based on the Randot stereoacuity test. Controls were age-matched to amblyopes. They showed normal or corrected to normal visual acuity in both eyes and significantly better stereoacuity compared to amblyopes. During experiment 1, we localized the motion selective areas in our subjects based on the response evoked by moving versus stationary concentric rings. You can find more details about our methods in this paper, recently published in the Journal of Cerebral Cortex. During experiment 2, we localized the stereoselective areas in our subjects based on the response evoked by 3D versus 2D stimuli, generated by random dot stereograms. Subjects saw the stimuli through red and green filters. More details can be found in our paper published in the Journal of Neuroscience, shown here. During experiment 3, we localized the color selective areas in our subjects based on the response evoked by color varying versus luminance varying stimuli. This test was an important control because we did not expect to see any impairment, or at least any strong impairment, in color perception in our amblyopes. Also, color selective sites could be used to better define the border of motion and stereoselective sites, as we have shown in our previous publications. Again, details of our methods are already published in multiple articles, including this one. Here you can see the location of those cortical sites that show significantly stronger response to moving compared to stationary stimuli in one control participant. Red to yellow colors indicate the p-value of 10 to the -2 to 10 to the -6. Borders of visual areas, indicated by the dashed black lines, were defined retinotopically based on separate scans and stimuli. The asterisk shows the location of the fovea. In the macro level, you can see area MT as the tip of the medial temporal sulcus, and this motion selective activity extends to area V3A. In finer scales, you can see stripy activity in V2 consistent with the expected organization of V2 stripes. This stripy activity extends to V3 and even V4. Here you can see the stereoselective sites in the same subject. Again, you can see stripy patterns in V2, V3, V3A, and V4. And then the color selective activity that starts from V1 and then in V2 stripes, extending to V3, then V4. Also, as expected, there is weak to no color selective activity in V3A. This slide shows the interdigitation of motion, stereo, and color selective sites in the same control individual. Red, green, and blue colors show the location of motion, stereo, and color selective stripes as you saw in the previous slide. Here you can see more examples of this phenomenon in 12 other hemispheres from our control participants. You can see the same phenomenon in area V3A, in which motion and stereoselective sites are detected with small overlap between them. In all cases, V3A is mostly occupied by stereo and motion selective clusters, while the color selective sites appear to be rare. Here you can see the motion selective activity in one amblyopic participant with no measurable stereo acuity. Similar to the control participant that I showed, you can see area MT, extension of activity to the V3A, as well as stripy activity in V2, and activity extending to V3 and V4. Here you see the response evoked by 3D versus 2D stimuli. As you can see, stereoselective sites are simply non-existent in this individual. This is something that we see in most amblyopes that show stereo blindness. Here you can see activity evoked by color versus luminance-varying stimuli. Again, similar to control subject that I showed, you can see strong activity in V1, V2 stripes, V3, and V4. Again, you can see almost no color selective activity in area V3A. Here you can see the interdigitation of these stripes. In almost all amblyopic participants, stereoselective sites are rare, but as in controls, interdigitated color and motion sites are easily detectable. You can also see the same thing in V3A. In most amblyopic participants, V3A is dominated by motion selective activity. Subsequently, we compared the size of motion, stereo, and color selective clusters between amblyopes and controls quantitatively. All measures are normalized relative to the size of area V2. In area V2, you can see a clear decrease in the number of stereoselective vertices. But interestingly, this effect was accompanied by an increase in the frequency of motion and color selective vertices, while the number of vertices that showed no selective response, including those that represented the location of the fixation object, was statistically equivalent between the two groups. Supporting this observation, an application of one-way repeated measures ANOVA yielded a significant interaction between effects of group and selectivity type. Similar results were also found in area V3. Consistent with the maps that we showed, area V3A in controls was mostly occupied by motion and stereoselective vertices. In amblyopes, we again witnessed a clear decrease in the number of stereoselective vertices, as well as an increase in the number of motion selective vertices. However, the change in the number of color selective vertices was comparably small. In contrast to V2 and V3, in V3A, we witnessed an increase in the number of vertices that showed no selective response. Interpretation of this effect is not very clear. It may suggest a more severe impact of amblyopia on V3A compared to earlier visual areas. It is also possible that these non-selective vertices have become responsive to more complex stimuli than simple radial motion, which we tested here. More tests are required before any conclusion can be made. Statistically, an application of one-way repeated measures ANOVA to the measured frequency of vertices with a group effect yielded a significant interaction between the effects of selectivity type and group. So, there are two main take-home messages. One is that amblyopia has a strong impact on the evoked stereoselective response. This is consistent with studies in animals that show a decrease in the number of stereoselective neurons due to amblyopia. In addition, we illustrated that color and motion selective sites are detectable in amblyopes like in controls. However, in V2 and V3, motion and color selective sites were larger in amblyopes compared to controls. In V3A, motion selective but not color selective sites were larger in amblyopes compared to controls. In conclusion, here we showed one of the first pieces of direct evidence of amblyopia's impact on the mesoscale functional organization of early visual areas using high-resolution fMRI. As expected from cortical plasticity, in absence of stereoselective response, motion and color selective sites appear to become larger in amblyopes' early visual areas, ruling out the possibility of neuronal degeneration in those regions. These findings raise an important question. Are amblyopes superior to controls in motion and color perception? The simple answer is we do not know. On one hand, clinical studies have already shown that many amblyopes suffer from impaired global motion perception, which is most likely linked to the function of motion selective clusters. There is also no reason to believe that having a larger neuronal cluster necessarily leads to a better function. On the other hand, there are studies like this one that suggest that poor stereo vision may have some positive consequences. One famous example of this is with Leonardo da Vinci. Through analysis of portraits and self-portraits, it has been suggested that da Vinci had strabismus. But even if we ignore the differences between amblyopia versus having poor stereopsis, without any doubt, it still takes a lot of training and practice to bring art out of an impairment. Here, we would like to first thank our participants as well as our collaborators and sponsors. Thank you all for your attention and have a good one.